Hi everyone, in this video we'll be continuing with our British Biology Olympiad preparation series, and we'll specifically be focusing on the biomolecule side of it, looking into carbohydrates specifically. Now, if you haven't done so already, I recommend that you watch part 1, 2, 3, and 4 of the series on carbohydrates because it contains a lot of prerequisite knowledge that's probably needed for the comprehension of the content that we will be dealing with in this video. Having said that, up to this point, we've looked at monosaccharides and disaccharides, and in this video, we'll be looking into polysaccharides, which are large carbohydrate molecules that are formed from many repeating sugar units. Now, polysaccharides are basically polymers, meaning that it's composed of a large number of monomers that are joined together. In carbohydrates, these monomers are going to be called monosaccharides, basically single sugar units, most commonly glucose. And these monosaccharide units are linked via glycosidic bonds, the sort of covalent bonds that are used in sugar, and this is going to be formed through condensation reactions, which are basically a type of anabolic reactions wherein we're taking away molecules of water in order to form a covalent bond to connect different monomers together to form polymers. Now, what's very crucial to understand is that polysaccharides with the same monomer can still have very, very different chemical properties, and the biological function of a polysaccharide depends on what it is made of, not necessarily only that, but also how the monomers are actually arranged and which glycosidic bonds are present, and what that does in turn to affect whether the structure is going to be branched or unbranched or linear. Now, starch is the main carbohydrate storage molecule that is used in plants, and plants synthesize starch as a way of storing excess glucose molecules that are produced during the process of photosynthesis in a form that is going to be insoluble, compact, and osmotically inactive. Most importantly, starch is not necessarily a single polymer. It's a mixture of two distinct glucose polymers, which is known as amylose and amylopectin. Now, both of the polymers are built exclusively from alpha glucose, but they do differ in structure, and the structural differences explain how starch functions to be such an effective storage molecule. We'll first be looking at amylose, the simpler of the two compounds. Now, amylose is a compound that's made entirely of alpha glucose molecules, and they're linked together by 1,4-glycosidic bonds. The bonds forms between carbon 1 of the first alpha glucose molecule and carbon 4 of the next. And most importantly, amylose is a linear, unbranched structure because we don't have any 1,6-glycosidic bonds present. And because every glucose unit is joined in the same way, the orientation of the molecule is highly consistent along the entire chain, and in particular, the hydroxymethyl group, or the CH2OH group, the orientation of this group on each of the glucose molecule present is going to all point to the same direction. Now, this consistent sort of orientation is going to cause the amylose chain to naturally coil in a helical sort of structure, and this helix makes amylose much more compact and relatively insoluble, which is very much advantageous for the storage purposes. It also explains why amylose produces a characteristic sort of color change in the iodine test, as iodine molecules fit within the helical structure. However, amylose is unbranched, meaning that it has relatively few terminal ends, and this limits the number of sites where the enzymes can act, meaning that amylose is broken down much more slowly than other branched polymers. Right now, we'll be moving on to the second component of starch, which is amylopectin, which is a much more complex structure. Now, amylopectin is composed entirely of alpha glucose molecules, much like amylose, but unlike amylose, it's going to have a branch structure. Now, along the main chains of amylopectin, glucose molecules are going to be joined by 1,4-glycosidic bonds, exactly as is in amylose. However, at certain points like these, a glucose molecule will form a 1,6-glycosidic bond, which is going to create this sort of branch. And each of the 1,6 bonds mark the start of a new chain that's branching off of the original chain. And these branches themselves are extended further through additional 1,4-glycosidic bonds, and it's going to essentially create what is a very highly branched tree-like structure. And branching has important functional consequences because it greatly increases the number of terminal glucose units, which is going to allow enzymes to act on many sites at the same time. And as a result, amylopectin can be hydrolyzed much more effectively and rapidly than in amylose when glucose is required. Now, the combination of compact storage and rapid accessibility seen in this specific compound, amylopectin, makes it particularly effective as a component of starch. And together, amylose and amylopectin allows plants to store large amounts of glucose in a very effective manner whilst still being able to release glucose when it is needed.